Welcome to the Rafikova Lab. I'm Joel James, a postdoctoral researcher in this lab, and I'll be taking you through a tour through this lab. Our research explores cardiovascular disease. I shall briefly explain few of our projects. The first project we do is to explore iron sulfur clusters and mitochondrial dysfunction. The mitochondria, as you know, is the powerhouse of the cell, and it is also very important in controlling the cell's metabolism. At this lab, we show that mitochondrial dysfunction can trigger pulmonary arterial hypertension, which is a cardiovascular disease and is quite lethal. We study the role of iron sulfur clusters in a mutant rat model, which we developed right here in this lab. Our second project focuses on gender-specific solutions in pulmonary arterial hypertension, or PAH. PAH is a disease with several causes and outcomes, and it is crucial to understand how males and females are affected. Our research has shown that males and females have different severity of the disease and also different outcomes. So we focus on understanding the mechanisms in males and females, and this would help in developing better therapeutics targeting the individual genders. Our third project focuses on the role of heme in PAH. Heme is literally everywhere in the body, and due to its association with hemoglobin, it is pretty much ubiquitous. So when heme is not associated with hemoglobin, it can be quite dangerous. We discovered that patients with PAH have a high correlation with free heme. What we do here at the lab is also to explore pathways that free heme can potentially trigger and which can possibly lead to pulmonary arterial hypertension. And our fourth project, what we do at this lab, focuses on a process called anaplerosis. In very simple terms, this is a metabolic process that can trigger sustained cell growth, and it can lead to excessive cellular proliferation. We show that with mitochondrial dysfunction, anaplerosis is activated, and this leads to PAH and uncontrolled cellular proliferation. So we aim to dissect mechanisms to understand how we can control anaplerosis. Our lab tackles these problems through several approaches. We use both rat and mouse models to study these problems. And we also study how we can design peptide inhibitors to control pulmonary arterial hypertension. We aim to transition our animal studies to the Petri dish, and we also collaborate with clinicians to work on human samples. We shall shortly see what sort of animal work happens in the lab, and we shall see this go to the surgery room. I think we are right on time to catch a surgery that's currently happening. Hi, Dr. Verghese. What's happening here?
So I'm doing a surgery on this mouse for checking the right ventricular systolic pressure. Here we can see the right ventricular systolic pressure, and this is measuring by inserting a catheter into the jugular vein, and this will tell us about the right heart pressure.
Okay, so what do you do after your surgery?
After this surgery, we will collect all the organs and blood for further biochemical analysis.
So, Dr. Matthews, what else do we do using the rat model?
This is the echocardiographic analysis of a rat, and this is the heart, it's pumping, and here we are using this echocardiography for studying the wall thickness, cardiac output, ejection fraction. This will tell us how heart disease is progressing and how it is normally functioning.
We have Dr. Maki Nihori here who would explain to us in brief what she does on rats.
I don't have an animals in front of me, but I have a really good pictures from the textbook how to expose the jugular vein, which is directly connected to the right side of the heart, which we are putting the catheter and to measure the right heart functions. So once the catheter goes into the right atrium, you will start seeing this noise in the measurement. Then if I push a little farther, you can get the good position, and you will get the right ventricle pressure in this monitor. As you can see, once we having a good position, we will get the really nice waves of the pumping of the heart and the changing the pressure in the right height.
So, what other technique do you use to measure heart function?
So we do have another catheter called the transonic PV catheters, and we can use this to measure the pressure and the volume at the same time. So we can do the pressure volume catheter and measuring the function of the right side of the heart as well.
Okay, and after your experiments, what do you do with the
So
rats?
I wanted to show you some samples that I collected after the surgery. So this sample was filled with silicones in pulmonary arteries after the surgery. Once I preserve the artery structure, I can get a really beautiful picture.
So these are lungs.
Yes, those are the lungs, and
And
those
the
are
heart.
Heart is here, and this is a lung. Yellow part is the silicone filled with the pulmonary arteries.
Okay.
After I collected those samples, I can do many things. The first thing we can do is take an image with a micro CT scan. This is the 3D images taken by the micro CT scan at the university. And once you get those pictures, you can see how the structure is and how the vessels is structuring in the lung directly.
Great. So what does the CT scan tell us in the end?
So we also has to get the angiograph picture like this. As you can see, we can find a lot of details at the tip of the pulmonary arteries, which is affected the most by the progression of the PAH. So if we get those data, we can compare how progressed the disease is, how we can attenuate the diseases at the same time with this technique.
Great. After Dr. Makini Hori does her surgeries, she gives me the heart samples, and I isolate cardiomyocytes from these hearts. And as you see here, the cells that are square and rectangular shaped, these are cardiomyocytes. And if you see, these cells are actually pulsating in the petri dish. So what I do is isolate a purer sample and then perform mitochondrial studies on these cells. This is our cell culture facility, and what we do here is to study the cells that we routinely isolate from the animal model. So we transfer the cells from in vivo to in vitro in a petri dish. This is a flow cytometer on my right, and we use this instrument to study the cell types, and we also use it routinely to count cells. It's a very crucial instrument at our lab. Right here is an incubator which holds the ECIS machine, and using the ECIS machine we can study how cells grow in real time. We can also study how cells adhere to each other and how the cells form a barrier. We can study barrier function using this particular instrument. We also use molecular biology techniques to manipulate cells. We do a lot of transfection, and if you're interested in molecular biology, this is probably a place for you to be. We saw what techniques we use at the lab to aid our discovery process, and I shall briefly run through all the instruments we have here. This is a ChemiDoc on my right, and this helps us carry out Western blots quite rapidly. And here, this room, we have a lot more instruments. We have a spectrophotometer, which is also a fluorescent spectrophotometer. It's quite useful. We have a HPLC, which helps us to do chromatography. And we also have a Bioplex, which is quite interesting. This helps us to analyze several cytokine markers in several samples at the same time, which gives us really good results in a very less time. And finally here we also have real-time PCR. So you've already seen the flow cytometer. We have techniques to do echocardiography. We can use, we have catheters to study pressure. So you've seen what we do at this lab. And what you can expect from this lab is to learn numerous techniques. And you'll also have a tremendous opportunity to advance your scientific career.